Question number 36. Study the following lists. List 1. A. Acidic amino acid. B. Basic amino acid. C. Aromatic amino acid. D. Neutral amino acid. List 2. 1. Tryptophan. 2. Lysine. 3. Valine. 4. Glutamic acid. 5. Methionine. So first one, A. Acidic amino acid. Acidic amino acid. So which one is called as acidic amino acid? Means the amino acid which contains more number of carboxyl groups, we call it as a acidic amino acid. From the given options, glutamic acid is acidic in its nature. B. Basic amino acid. So basic amino acid means the amino acid which contains more number of amino groups in it. So such type of amino acid will be basic in its nature. So from the given options we can say that B 2, that is lysine is as alkaline in its nature or basic in its nature. C. Aromatic amino acid. Aromatic amino acid means the one which is having the aromatic nature or circular nature, that is tryptophan 1. Then neutral amino acid. Neutral amino acid means the amino acid which contains nearly equal number of amino groups and the carboxyl groups in them. So such type of amino acids are called as neutral amino acids. The example for neutral amino acids is valine. So A is 4, B is 2, C is 1 and D is 3. So correct option is 4, 2, 1 and 3. Question number 36, the correct answer is 4. Question number 37. Study the following lists. List 1. A. The number of CN rings present in 360 degrees angle of double-stranded DNA. B. The number of CN rings present in the 180 degrees angle of double-stranded DNA. C. The minimum number of hydrogen bonds present in 360 degrees angle of double-stranded DNA. D. The number of phosphodiester bonds present in 360 degrees angle of double-stranded DNA is. List 2. 1. 40, 2. 15, 3. 18, 4. 20 and 5. 30. Okay. Let's come to the list 1. A. A is related to the number of CN rings present in 360 degrees angle of double-stranded DNA molecule. So in a 360 degrees angle means, so this is one coil of DNA molecule, and as we know that one coil of DNA molecule contains 20 nitrogen bases. Out of these 20 nitrogen bases, 10 will be purines and remaining 10 will be pyrimidines, because one of the purine is complementary to the other one. So that's the reason why here the number of purines are 10 and the number of pyrimidines are also 10. In this each purine is an dicyclic compound. So it contains each of the purine contains 2 carbon nitrogen rings. So 10 into 2, a total of 20 carbon nitrogen rings are present. When it is coming to pyrimidines, each pyrimidine is a monocyclic compound. That means each pyrimidine contains only one carbon nitrogen ring. So 10 into 1, it comes to 10 carbon nitrogen rings. So the total number of carbon nitrogen rings which are present in 360 degrees of angle of a double-stranded DNA is 30. So in the given options, so which one is more related means option number 5. So A is 5. Then the second one is B. The number of CN rings present in 180 degrees angle of double-stranded DNA molecule. Okay, here 180 degrees of DNA molecule means it is having half coil, and half coil contains 10 nitrogen bases. Out of these 10 nitrogen bases, according to the complementary nature, 5 will be purines and 5 will be pyrimidines. So 5 purines are present, and each purine contains 2 carbon nitrogen rings. So 5 into 2, 10 carbon nitrogen rings are present in 5 purines, and the remaining are 5 pyrimidines. Each pyrimidine is having 1 carbon nitrogen ring, so that 5 into 1 carbon nitrogen ring in each of the pyrimidine. So total number of carbon nitrogen rings in 180 degrees of double stranded DNA is 15, that is 10 plus 5, which is equals to 15. From the given options, B is 2. Then C, the minimum number of hydrogen bonds present in 360 degrees angle of double stranded DNA. So once again here 360 degrees of DNA molecule means which is having one coil, and if all these nitrogen bases which are present in this one coil are of only A and T, then there is a possibility of a minimum number of hydrogen bonds in it. So for example, if all the 10 base pairs are of only A and T, so how many base pairs we are saying? 10. So 10 base pairs into each base pair contain 2 hydrogen bonds between them when they are only A and T pairs. So 10 into 2, 20 base pairs. Okay, 20 hydrogen bonds are there for this 10 base pairs. So from the given options we can say that C is exactly fitting to option number 4. Then the last one, that is D. Then the number of phosphodiester bonds present in 360 degrees angle of double stranded DNA is. Okay, here as we know that DNA is having two strands, or it's an polymer of nucleotides, and 2 such polynucleotide strands are present, and on one side 10 phosphodiester bonds are present. Okay, here if you take the diagram here, so here 10 sugar molecules on one side, then each of them is binded to one phosphate molecule like this. These are the phosphate molecules. Then this is a nitrogen base. We can take any nitrogen base here. 10 nitrogen bases on one strand. So here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 bonds, 10 nitrogen bases are there. This is related to one side of the strand. And if you take the other side of the strand, so sugar molecules, 10 sugar molecules we had to show, that is pentose sugars, and each of them is binded to the phosphate molecules in this manner. Okay, like in this pattern here each of them is binded to one nitrogen base. Okay, now each of this, for example, if this is A and T, if this is G and C, like this 2 bonds and 3 bonds, if this is not concerned with our question here, we are concerned with the phosphodiester bonds. So where are these phosphodiester bonds are there means these are the phosphodiester bonds which are present between the pentose sugar and the phosphate molecule. Okay, on one side, okay, here if you are taking, this is the 2 strands that have been given here, these are the 2 strands that have been given here, we have to consider one side of this one. So that is 1 phosphodiester bond, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17 and 18. So on one pitch we can see the presence of 18 phosphodiester bonds. So D is 3. D is 3. So here one coil which is having the 18 phosphodiester bonds on one side. So here from the given options, A is 5, B is 2, C is 4 and D is 3. So question number 37, the correct answer is 1. Question number 38, assertion, starch exhibits a helical secondary structure. Reason, starch iodine combination is blue as starch can hold iodine molecule. Okay, here starch is an polysaccharide molecule which is present in plant cells. This is a molecule which is is a part of biomacromolecules, and starch is exhibiting the helical secondary structure, so that statement is correct. And starch have one more ability, that it can bind the iodine molecule because it is having helical structure, and because of this combination between starch and iodine, it is exhibiting blue color also. So reason statement is also correct, but so first one is talking about the structure of that starch molecule, second one is related to the presence of iodine and helical nature. So both A and R are correct, but R is not a correct explanation for assertion. Question number 38, answer is 2. Question number 39, assertion, living organisms exhibit a wide diversity in biosphere. Reason, all living organisms are made up of same chemicals. Okay, assertion statement is nothing but that biosphere means the surface or the earth on which the place where the plants or animals or other type of living organisms can survive. That means it's a wide diversity that we can see in the biosphere. So that statement is correct. And all living organisms are made up of same chemicals. That is also correct. And one more important thing, that the composition of all the living organisms is not exactly same. There is a slight difference may be there, and the composition of each and every element may be slightly differing from one living organism to the other living organism. So the reason that have been given is correct, but reason is not the correct explanation for assertion statement. Question number 39, the correct answer is 2. Question number 40, assertion, both primary and secondary metabolites are synthesized in the protoplasm of the same cell. Reason, the functions of primary metabolites are known, whereas most of the secondary metabolites are unknown. Okay, let's come to the first statement, that is both primary and secondary metabolites are produced inside the cytoplasm of the same cell. Okay, here primary and secondary, both of them are the organic molecules which are synthesized in the cytoplasm of the same cell, but they are differing in their functions and their identity of the functions also. Why? Because a primary metabolite is a substance or a chemical which is having a defining, or we can say that an identifiable function inside a cell, and secondary metabolite is also an organic molecule which is not having a identifiable function as like that of a primary metabolite. So that is the difference that we observe between the primary and the secondary metabolite. So that based on this explanatory part, we can say that assertion is correct and reason is also correct, but assertion is not being explained by the reason statement. Question number 40, the correct answer is 2. Question number 41, assertion, adenine and guanine are dicyclic and homocyclic ringed molecules. Reason, thymine and cytosine are heterocyclic and monocyclic ringed molecules. Okay, let's come to the first statement, that is assertion. Assertion is saying that adenine and guanine are dicyclic and homocyclic ringed molecules. Actually, adenine and guanine are purines. Purines are dicyclic, correct. That is they contain two ring like structures, so that is correct. and the two rings which are present are not similar to each other. That's the reason why these are heterocyclic ringed molecules. But in the assertion statement it is given as homocyclic. That part is incorrect. So assertion statement is wrong. Then coming to reason statement. Thymine and cytosine, both of them are pyrimidines. These are heterocyclic compounds. Correct, because each of them is made up of two different types of elements, that is carbon and nitrogen. So that is correct. And these are monocyclic ringed compounds. That is also correct, because they are made up of only one ring structure. So both thymine and cytosine are thymine and cytosine are monocyclic and heterocyclic compounds. So reason is correct. Question number 41, correct answer is 4. Question number 42, which of the following components of nucleic acids are dicyclic and heterocyclic molecules? 1. Adenine, 2. Thymine, 3. Cytosine, 4. Guanine, 5. Uracil. So let's come to first one, adenine. 2. Thymine, 3. Cytosine, 4. Guanine, 5. Uracil. Okay, out of these two, out of these 5 that have been given here, adenine is an purine molecule. So it is dicyclic and heterocyclic molecule. Thymine is pyrimidine. It is monocyclic and heterocyclic compound. Cytosine is also pyrimidine. So it is also monocyclic and heterocyclic compound. And guanine is purine. This is dicyclic and heterocyclic compound. When it is coming to uracil, uracil is a pyrimidine which is present in RNA molecule replacing thymine. So as it is a pyrimidine, it is also monocyclic compound and heterocyclic in its nature. So which of the following nucleic acids are dicyclic and heterocyclic molecules? So dicyclic and heterocyclic molecules are 1 and 4, that is adenine and guanine. Question number 42, the correct answer is 3. Question number 43, select the mismatch from the following. 1. Cytidine, nucleotide of cytosine. 2. Guanosine, nucleoside of guanine. 3. Uridine, nucleoside of a pyrimidine. 4. Adenine, nitrogen base of DNA only. 5. Adenosine, nucleotide of adenine. 6. Thymidyllic acid, nucleotide of a pyrimidine. So from that we have to identify the incorrect matches. So first one is cytidine. Cytidine is a nucleotide of cytosine, nucleotide of the cytosine that have been given here. So here actually cytidine is a nucleoside of cytosine, not nucleotide. It is a nucleoside of cytosine. But in the option that is given as a nucleotide of cytosine. So second one is guanosine. Guanosine is a nucleoside of guanine. So in the option they had given the correct one, that is nucleoside of guanine. So this is given correct. Then third one is uridine. Uridine is a nucleoside of a pyrimidine that have been given here. So actually uridine is a nucleoside of uracil, and uracil is a pyrimidine. That is correct. Then fourth one, adenine. Adenine is an nitrogen base that is present both in DNA and also in RNA. But in the option they had given that nitrogen base of DNA only. Only is a wrong notion that is given here. So that it's a nitrogen base of both RNA and DNA molecules. So it is given wrong there. Then fifth one is adenosine. Adenosine is the nucleoside of adenine, but in the option they had given it as nucleotide of adenosine. Okay, nucleotide of adenine. So actually it is nucleoside of adenine molecule. Then sixth one that have been given is thymidyllic acid. When you are saying acid, that represents the nucleotide nature, because it includes the phosphoric acid in it. Phosphoric acid in it. So here it is a nucleotide of thymine molecule. Thymine molecule. Thymine is a pyrimidine. So this is correct. This is correct. So from the given options, which one of the mismatch means 1, 4 and 5 are the mismatches that have been given in the table. So these three mismatches are there in the third option. So question number 43, the correct answer is 3. Question number 44, the genetic nucleic acids are options 1 DNA, 2 RNA, 3 nitrogen base, 4 pyrimidines. Okay, from the given options, here it is saying that genetic nucleic acids, that is the nucleic acids which are acting as genetic materials. We know that most of the organisms have DNA as genetic material, which is helping in the transfer of characters, or they are helping in inheritance, okay, from one generation to the another generation. So DNA is the most frequently usable form of DNA genetic material in most of the living organisms. And when it is coming to RNA, RNA is also acting as genetic material, but only in few organisms. That to be in some of the plant viruses, RNA is acting as genetic material. Then coming to nitrogen bases. Nitrogen bases are not an individual genetic materials, and also in the case of pyrimidines. So from the given options, DNA and RNA are acting as a genetic nucleic acids. So question number 44, the correct answer is 1. Question number 45. The repeating monosaccharide present in cellulose are options 1 fructose, 2 glucose, 3 sucrose, 4 lactose, 5 galactose. Okay, here glucose is a molecule which is a monomer present in cellulose, and no other monomer is present in cellulose apart from glucose molecule. That means from the given options, glucose is a only monomer which is repeatedly present in the cellulose part, and otherwise we can say cellulose is the main chemical which is present in the cell wall of many plants, or maximum number of plants, which is an polymer, or we can say homopolymer of glucose molecules. So question number 45, the correct answer is 3. Question number 46. Study the following statements. 1. Metabolic reactions do not occur in isolated form, and they are always linked to some other reactions. 2. A series of linked reactions are called metabolic pathways. 3. The metabolic pathways are either linear or circular. 4. there is no uncatalyzed metabolic conversion in living systems. The correct statements are first one 1 and 3, second one 2, 3 and 4, third one 1, 2, 3 and 4, fourth one 1, 3 and 4. Okay, let's come to the options that have been given here. So the metabolic reactions do not occur in isolated form, and that is very much correct, because always one metabolic reaction is linked somewhere with some other metabolic reaction which is taking place inside the cells. So that means they are not in isolated form, and they are always interlinked with some other reactions at one place or the other place. So first statement that have been given is correct. Then coming to the second one, a series of linked reactions are called as metabolic pathways. So here we said that all the reactions which are taking place inside a living cell are not isolated, they are linked with each other, and all that reactions which are there and linking with each other, all that entire process we call it as a metabolic pathway. So statement 2 is also correct. Then third statement, the metabolic pathways are either linear or circular. So the reactions which are taking place in a living cell may be going in a linear fashion. That is, linear means one first compound, second to third, without any branching like structures. And sometimes they may be in circular form also. Circular form means some of the products which are formed in the metabolic reactions may be participating in some other reactions, and those are once again will be coming to the initial substrates or products. So that is, we can say that they are interlinked and they may be circular also. So statement 3 is also correct related to the question. Then there is no uncatalyzed metabolic conversion in living systems. Actually, we know that many chemical reactions which are taking place inside the plant cell and also in the living animal cells and in all living systems, and each and every reaction is catalyzed by some specific enzyme molecule. That means there is no such chemical reaction in a living system where enzyme is not participating. So there is no such uncatalyzed reaction in a living system. So that is also correct. From the given options, we can say all the 4 statements that have been given, all these are related to the metabolic reactions or the pathways which are taking place inside a living system, and all the statements are correct. Question number 46, the correct answer is 3. Question number 47, in plants, cellulose molecules are generally found in 1. Cell wall, 2. Paper made from plant pulp. Paper made from plant pulp. And third one is cotton fibers. Cotton fibers. From this, we have to identify which of the following structures are generally made up of cellulose in a plant body. Cell wall. We know that presence of cell wall is a unique feature present in plants. Why we are saying so means this character is present only in plants but not in animals. And cell wall is made up of mainly by a chemical called as cellulose. Apart from pectin, here cellulose is the other main compound. So that related to that plant, it is correct. Paper made from plant pulp. Here we are saying paper. Now the paper which we are saying is not related to the plant. It is not present in the plant now. That's the reason why it is not the correct combination. Then cotton fibers. So here we know that the cotton seeds will produce some epidermal hairs. Those hairs are called as a cotton fibers, and as these are the extensions of the epidermis, the cell wall which is present in the epidermal cells is present in the cotton fibers also. That's the reason why the cotton fibers are also made up of cellulose material. So from the given options, we can say that cellulose is present in the plant parts like cell wall and the cotton fibers. So question number 47, the correct answer is 2. Question number 48. Glycosidic bonds form between 1. individual monosaccharides in a polysaccharide. 2. Nitrogen base and pentose sugar of nucleic acids. 3. 2 polynucleotide chains of nucleic acid. 4. 2 pentose sugar molecules and a phosphate molecule. Okay, let's come to the first option. We know that polysaccharides are polymers of monosaccharides, and these monosaccharides are binded with each other with the help of an bond called as glycosidic bond, and this glycosidic bond is formed and a water molecule is released from that one, that is by a process called as dehydration process. So that means this one is correct related to the presence of glycosidic bond. Then the second one is nitrogen base and a pentose sugar of nucleic acid. We know that in a nucleotide of a DNA molecule, this is a pentose sugar, and we know that this is a 5 carbon molecule. We are not concerned with the attachment to the phosphate here. We are mainly concerned with the first carbon of pentose sugar is binded to a nitrogen base, and the bond which is present between the first carbon of the pentose sugar and the nitrogen base is a glycosidic bond. That means the second one is also correct. Third one, 2 polynucleotide chains of nucleic acid. 2 polynucleotide chains means the 2 strands of DNA that are present, those are binded to each other with the help of hydrogen bonds. Those hydrogen bonds are present between the nitrogen base of one strand and the complementary nitrogen base of its opposite strand. So it is not by the glycosidic bond, but it is with the help of hydrogen bonds. So third one is incorrect. Fourth one, 2 pentose sugar molecules and a phosphate. Already we had studied that each pentose sugar in a DNA molecule or in a RNA molecule is binded to 2 phosphate molecules, one on the 5th carbon and other on the 3rd carbon. And in that formation, we can say that the bond which is present between 5th carbon and the phosphate is one bond, that is ester bond, and one more that is present between the 3rd carbon and the phosphate is also one ester bond. So one pentose sugar is having 2 bonds, so we call it as a phosphodiester bond. So that is the bond which is present between the phosphate molecule and the sugar molecule, but not the glycosidic bond. So that means fourth one is also incorrect. So from the given options, we can say that 1 and 2 are correct and 3 and 4 are incorrect. So we have to identify the correct combinations here. So question number 48, the correct answer is 4. Question number 49. In which of the following dehydration reaction occurs? 1. During peptide bond formation. 2. During phosphodiester formation. 3. During glycosidic bond formation. 4. During hydrogen bond formation between nitrogen bases. So the correct options or the incorrect options that have been given are 1 and 4, 2 and 4, 3 1 and 3, 4 1, 2, 3 and 4. Okay, let's come to the first one, that is dehydration is taking place during peptide bond formation. So peptide bond is formed between the 2 amino acids which are present adjacent to each other, and when the peptide bond is formed between COOH of earlier amino acid and NH2 of the next amino acid, one water molecule is released. So that is by a process called as dehydration process. So that is correct. Second one is during phosphodiester bond formation. Phosphodiester bond is present between the pentose sugar and the phosphate molecules of DNA molecule or RNA molecule. That bond is formed not by releasing of a water molecule, that is, it is not because of the dehydration process. So that is incorrect. Then Then coming to third one, during glycosidic bond formation. Here this glycosidic bond is related to the bond that is formed between monomers of a polysaccharide molecule. Okay, here the glycosidic bond which is formed between the monomers of a polysaccharide is by a loss of water molecule. So that is also a process called as dehydration. So that is correct. Then the fourth one, the hydrogen bonds which are present between the nitrogen bases. Okay, here in that during that process the water release is not taking place. So dehydration is not taking place. So that is also incorrect. So from the given options we can say that 1 and 3 are correct. That is dehydration is taking place during the peptide bond formation in the protein chain and glycosidic bond formation in a polysaccharide molecule. So question number 49, the correct answer is 3. Question number 50, study the following statements with reference to chitin. Options that have been given here. 1. It is present in animal cell walls. 2. It is present in exoskeleton of arthropoda. 3. It is a heteropolymer. 4. Amino sugar and other modified sugars are its building blocks. Okay, here let's come to the correct statements. The correct statements are: first one 1, 2 and 3, second one 2, 3 and 4, third one 1, 2, 3 and 4, fourth one 2 and 3. Okay. Now the first one is chitin is a special type of heteropolymer molecule which is present in the exoskeleton of arthropods but not in the all animal cell walls. That's the reason why the first statement is incorrect. And as already said that it is present in the exoskeleton of the what you call arthropods, that is insects. Because of that chitin, the exoskeleton is hard in its nature. So that is correct statement. Then third one is the chitin is an heteropolymer and chitin is a nitrogen containing polysaccharide molecule. Polysaccharide, it is an heteropolymer molecule, that is it is made up of different types of monomers, or we can say that monosaccharides. Different types of monosaccharides are there to form into a heteropolymer molecule called as chitin. So 3 is correct. Then as we know that it is a modified sugar like amino sugar, that is amino sugars and other modified sugars are its building blocks. So it is made up of monomers, we said no, those monomers are nothing but the amino sugars and other modified sugars which are forming the building block like structures and giving rise to a heteropolymer called as chitin molecule. So this is also correct. So from the given options we can say that 2, 3 and 4 are correct related to the chitin molecule. Question number 50, the correct answer is 2. Question number 51, read the following table and identify the incorrect combinations. So here 3 combinations have been given: nitrogen base, nucleotide and nucleoside. Okay, under that first one adenosine, adenine and adenylic acid. Second cytosine, cytidylic acid, cytidine. Third guanine, guanylic acid, guanosine. Fourth thymidine, thymidylic acid and thymine. First one 1, 2, 3 and 4, second one 1 and 4, third one 2 and 3, fourth one 1, 2 and 4. Okay, now let's come to the question part here. They are given the nitrogen base in the first column. Second column is a nucleotide and the third column is a nucleoside. So nucleoside. Now let's come to the first one. They had given adenosine. Actually adenosine is not a nitrogen base but it is a nucleoside. It's a nucleoside and that means it is wrong. And adenine. Actually adenine is not a nucleotide but it is a nitrogen base. So this is also incorrect. adenylic acid. We are saying acid. Adenosine plus phosphoric acid is equals to adenylic acid. So, we are saying adenosine is a nucleoside plus phosphoric acid that forms into a nucleotide called as adenylic acid. But in this question they had given it under nucleoside. That means it is also incorrect. Second one, cytosine under nitrogen base. Okay, this is correct because cytosine is a type of nitrogen base which is pyrimidine. And second one, cytidylic acid. As I said that when we are saying acid, it should be a nucleotide because nucleoside plus phosphoric acid. Here as we are saying acid, it is a part of nucleotide, so it is correct. Then cytidine have been given under nucleoside. Cytidine is a type of nucleoside which is formed by the combination of cytosine and pentose sugar. So, this is also correct. Then third one is guanine. Guanine is a type of nitrogen base which belongs to purine. So, that is correct. Second one that have been given is guanylic acid. And as it is a acid, that is guanosine plus phosphoric acid. Guanosine is a nucleoside plus phosphoric acid forms into a nucleotide called as guanylic acid. So, this is correct. Then third one is guanosine in this one. Guanosine is formed by a guanine molecule and a pentose sugar molecule. So, guanine is a nitrogen base plus a pentose sugar. It is a type of nucleoside. So, guanosine is a nucleoside. It is correct. And the fourth one is thymidine. Thymidine is a type of nucleoside, but it have been given under nitrogen base. Thymidine is formed by a thymine molecule and a pentose sugar molecule. So, that it is incorrect. Then thymidyllic acid. Thymidyllic acid means it is an acid which is formed by combination of a nucleoside, that is thymidine plus phosphoric acid. So, this is correct. Then thymine. Actually, thymine is a nitrogen base, but it has been given under nucleoside. So, it is also incorrect. From the given conclusion part, we can say that only 2 and 3, all the combinations in these two are correctly matching to the nitrogen base, nucleotide and the nucleoside part. So, here question number 51, the correct answer is 2. Question number 52, read the following table and identify the correct combinations. Here 3 lists have been given. List 1 contains nitrogen base, list 2 contains group, list 3 contains number of CN rings. Okay, first one adenine, purine 2. Second one guanine, purine 2. Third one thymine, pyrimidine 1. Fourth one cytosine, pyrimidine and 2. Fifth one uracil, purine and 2. Okay, the options that have been given are first one 1, 2 and 3, second one 3 and 4, third one 4 and 5, fourth one 3, 4 and 5. Okay, now let us come to the correct matches to see whether the matches that have been given are correct or not. Now, first one is a nitrogen base, the second column is a group, and the third column is the number of CN rings. Number of CN rings. The first one adenine. Adenine is a purine molecule, that means it belongs to purine. And as we know that all purines contains 2 carbon nitrogen rings, because these are heterocyclic and dicyclic molecules. and whatever the options that have been given are correct in the question. So this is correct. The second one is guanine. Guanine is a nitrogen base which belongs to purine group. So it is also correct, that correctly given in the options. And as it is a purine molecule, it also contains 2 carbon nitrogen rings because it is a dicyclic compound and a heterocyclic compound. So this is also correct. Then third one is thymine. Thymine is a type of pyrimidine molecule. So it belongs to group called as pyrimidine. And as it is a pyrimidine, it contains only 1 carbon nitrogen ring. It is a heterocyclic compound, but it contains only 1 carbon nitrogen ring. That's the reason why the number of carbon nitrogen rings are only 1 here. So here whatever the options that have been given in the question are correct. Then fourth one, cytosine. Cytosine is a nitrogen base which belongs to a group called as pyrimidine. And all pyrimidines contains only 1 carbon nitrogen ring. But in the question, in the options that have been given as 2. So here that point is incorrect related to this one. Actually it should be 1, but it is given as 2. Then fifth one is uracil. Uracil is a pyrimidine molecule which is present in RNA. So, but in the question it is given as purine molecule. So this is incorrect. It should be pyrimidine. And as it is a pyrimidine molecule, it should contain only 1 carbon nitrogen ring. But here it is given as a 2. So this is also incorrect. It should be 1. So from the given options, we can say that only 1, 2 and 3 are correctly given and correctly matched. So question number 52, the correct answer is 1. Question number 53, the length of double stranded DNA is 340 angstroms. It contains 20% cytosine. What is the total number of hydrogen bonds found between purines and pyrimidines? Options 1 320, 2 120, 3 560 and 4 240. Okay, we know that some basic information related to the one coil containing 34 angstroms length, and that one coil contains 20 nitrogen bases or 10 base pairs. So one coil with 34 angstroms length and having 20 nitrogen bases, or we can say that it contains 10 base pairs. Now in the question they had said that the length of the DNA molecule is 340 angstroms. 340 is 10 times to that of 34 angstroms. So the number of coils that are formed by this DNA molecule is 10 coils. When it is forming 10 coils, so 20 nitrogen bases into 10 in each, so that it comes to 200 nitrogen bases. The total number of nitrogen bases that are present in the DNA that have been given in the question is 200. Okay, then this 200 nitrogen bases is equals to 100%, because whatever the number of nitrogen bases present in a DNA, that is equal to 100%. Now one more clue is given in the question, that is cytosine is 20%. Out of this 100%, cytosine is 20%. When cytosine is 20%, we know that it is complementary to guanosine or guanine, so it is also 20%, which comes to 40% out of this 100%. Then 20% of 200 is 40. So the number of cytosines are 40, number of guanines are also 40. And how many pairs of C and G are present? Here C and G pairs are 40. Between one C and G, we know that 3 hydrogen bonds are present. So 40 pairs into 3 hydrogen bonds. It comes to 120 hydrogen bonds. Then there is one more part which is left out here, that is the 60% of the DNA molecule. The 60% contains adenine and thymine, and both of them are in equal proportion. So adenine will be 30% and thymine will also be 30%, comes to 60%. And 30% of 200 nitrogen bases is 60. The number of adenines are 60, the number of thymines are also 60, it comes to 120 adenines and thymines. And how many pairs of A and T are there? A and T pairs are 60 pairs. And each pair contains 2 hydrogen bonds. That's the reason why 60 into 2. The total number of hydrogen bonds which are formed by 60 pairs of A and T are 120. So already we got 120 in CG. So 120 formed by C and G pairs and 120 formed by A and T pairs. The total number of hydrogen bonds that are present in a DNA molecule which is having 340 angstroms length and 20% of cytosine is 240. So question number 53, the correct answer is 4. Question number 54, the ratio between sugar and phosphate molecules in a dinucleotide is options 1, 2:1, 2, 1:2, 3, 1:1 and 4, 3:2. Okay, here we are saying dinucleotide. Whatever may be the nucleotide, the proportion between the sugar and phosphate molecules will be only 1:1. There will be no change in the ratio here. So question number 54, the correct answer is 3. Question number 55, assertion. Both prokaryotic and eukaryotic organisms have similar type of purines and pyrimidines. Reason, in nucleic acids, the purine and pyrimidine types are in 2:3 ratio. Okay. Here we know that in the nucleic acids, purines and pyrimidines are present. Purines include adenine and guanine. Pyrimidines include that is cytosine, thymine and uracil. Whatever may be the type of living organism, there will be no change in the type of purines and pyrimidines. That is, these are universally same in all types of living organisms. So the statement that have been given in assertion is correct. Then in nucleic acids, purines and pyrimidines are purine and pyrimidine types are 2:3 ratio. Here let's see the purines. Purines are adenine and guanine, whereas when it is coming to pyrimidines, pyrimidines are 3 types. Those are cytosine, thymine and uracil. So here the question is related to nucleic acids. When it is related either to DNA or RNA, the pyrimidines number will be 2. But as it is related to nucleic acid, the number of pyrimidines will be 3. So the ratio between the types of purines and pyrimidines is 2:3. So that reason statement is also correct. But whatever the statement that have been given in assertion and reason are nowhere related to each other. So both A and R are correct. R does not explain A. Question number 55, the correct answer is 2.